Can I see that?
Animals, what are the other two?
Humans
Humans?
and he's not animals, humans, and
You always get the last. Not plants as much.
no, isn't it the environment?
Huh?
Isn't it the environment like
It is, but what's the term they use for that?
Dang it.
It's so basic, it's probably gonna
Yeah.
go crazy. Did I show you the antibiotic resistance thing that I wanted to show you the other day in lab? The-
Non-living.
Non-living, there you go. I saw it right here. Yeah. Okay, I just saw it right here. I didn't know if I showed it to you or not. All right, why are they important? Why are reservoirs important?
Because they can hold on to disease and make you sick,
It makes it virtually impossible to do what?
cure things.
Not cure. You can cure disease, eradicate, yeah. So there's
Oh.
a different guy. Don't confuse cure with eradicate. Cure, I can cure E. coli, right? I can cure salmonella. But am I ever going to get rid of that? No. Why are we able to get rid of smallpox? What allowed us to do that?
Because of the resistance from the cowpox.
Nope. Why are we able to eradicate? We were just talking about that. Why are we able to eradicate? Officially, the WHO and everybody else has said that smallpox has been eradicated
Completely?
completely. They're-
Vaccinations, maybe?
Because, yeah, why do vaccinations work?
Because it prevents the disease from being passed around. Like it's preventing the reservoirs.
It does, but there is no reservoir. That's the point. Is there was no non-human reservoir. Since it was only human disease, once you vaccinate enough people, you got rid of it.
Yes.
All right. Can we get to this part? Is this where we stopped? And we went through all this, right? Patients that ignore pathogens that are ignored as potential causes of disease. For a long time, did they know that HPV caused cervical cancer?
No.
No, so it would have been ignored for a long time, right? That's what they're talking about, things like that. I talked about toxoplasmosis. We know what it does to developing fetuses. How has it been ignored? How was it ignored for years in adults? What do they link it to?
To causing problems in pregnancy?
That's for the developing fetus.
Oh.
But they didn't think there was a problem with it in adults, even though most people have it. Most people have been exposed to toxoplasmosis. What did I say was that link? They tentatively linked it to. They tentatively linked it to schizophrenia.
Schizophrenia.
And if you've ever seen mice that are infected with toxo, you seen that with them? Anybody ever seen that? They lose their fear of
Oh,
cats.
you did say that.
So they'll have an infected mouse and they'll have a cat right here and that mouse will just sit there and nothing happens. And it's because it affects the brain. It's pretty freaky to watch. Okay. Pathogenicity. We didn't go through these? Yeah. All right. The ability of a microorganism. Pathogenicity is the ability of a microorganism to cause disease. Can it make someone sick? Virulence is the degree of pathogenicity. Sometimes these two are kind of hard to tease out a little bit. Because virulence, a more virulent disease is going to more what?
Spreadable, isn't it?
But more easily cause disease. Okay, we talked about BSL1, BSL2 levels. Remember that? So if you have a BSL2 organism, it's probably going to be less virulent than a what?
3 or 4.
Than a BSL3 or something like that. Can E. coli make you sick?
Yes.
Yeah, you get the wrong strain, it can make you sick, it can make you pretty sick. Can Shigella make you sick?
Yes.
Which one's more virulent?
Shigella.
Shigella. So there got some of these virulence factors. So the degree of pathogenicity, and we're going to look at different ones. Adhesion factors, we already talked about these, right? What are they? What are the two we talked about?
Was that
One's a structure, the other one's more of a,
Slime layer.
part of it's the slime layer.
Fimbriae?
And what are the adhesion ones? The anchors I talked about. Now we went over. Where is it? Right here. Remember this too? Adhesion proteins, remember those?
Yeah.
So E. coli has these little live ligands on the surface of the bacteria. That ligand is going to interact with a particular cell surface receptor to allow it to attach. And that's critical for what type of organism. If without this ability to attach, no organism would be non-pathogenic. Or in other words, what organism is it actually critical that it can have these attachments to receptors on cells? Which one?
Maybe ones that hit your blood cell like
It's a particular type of disease-causing organism. Which one? Which ones absolutely have to be able to interact with the cell?
viruses.
Viruses. That's how I was going to viruses, guys. All right, let's get back to this part. Okay, so biofilms, that's going to be slime layers and things that can become biofilms. Extracellular enzymes, toxins, and antiphagocytic factors. And these two are going to be different. A lot of people think they're the same thing, but they're both excreted by the bacteria.
They're both excreted?
And we'll go through them one at a time. So here's Koch's postulate again. So what is Koch's postulate telling us? You've got 3 different mice. You had to be able to do what? Under Koch's postulate. What did they isolate from those 3 different mice? And what did they not isolate from this particular healthy one?
The bacteria that's causing.
This particular red bacteria right here. It's right here on this plate, here on that plate, here on that plate. Does it have it on this plate?
No.
So did they isolate that bacteria from that healthy animal? They grew it up, they identified it, struck it out on plates, re-inoculated a healthy individual,
Oh.
and they killed Mickey again. We take Mickey out. Mickey dies, but what do you have to do to confirm Koch's postulate?
Test it, pull it out again.
You had to be able to re-isolate that same organism from the test animal, and that's what they did. So what does that tell you? What did this whole experiment tell you right here?
That it's a pathogen that
Not only
can
is
be
it a
treated.
pathogen, but what did it tell you? What does Koch's postulate tell you?
What is that in the injection? Like more bacteria? Or
This was this bacteria right here.
Oh, okay.
So what does that tell you? What does Koch's postulate tell me right here? This experiment.
The specific pathogen, isn't it?
It tells you that this pathogen caused the disease in these mice.
Yeah.
This is what caused them. So now you can say, whatever this pathogen is, the suspected agent is the one that causes this disease. So bacteria X caused disease X. All right, and this is in terms of virulence. Lactobacilli and Dipteroides are not very virulent. Now, this had better not be because this is one of the what?
That we have in our body.
And it's also in what a lot of people take every day. This is one of those probiotics, so you don't want this to be very virulent, all right? Dipteroides is a little bit different. The Candidus albicans, vaginitis and thrush, that's a fungal infection, a yeast infection, but when does it cause disease?
With immunocompromised?
When you're immunocompromised somehow. It's not going to be very virulent, it's not going to cause disease on its own in a healthy individual. Now you got C. diff, Clostridium difficile. This is in everybody's colon, but it's supposed to be there. It's a commensal, right? When does it cause disease?
When it overgrows your flora.
Good. You get antibiotic-induced colitis. You take too many antibiotics, you lose that competition, C. diff comes flaring up, and that's when it can cause problems. Pseudomonas. This is one that affects who? Two
Burn
people.
victims.
Burn patients. What are the 2 acute concerns for third-degree burn patients?
Dehydration.
And infection. In particular, you worry about Pseudomonas. Normally, it's not that infectious. Everybody here probably has Pseudomonas on them right now. It's usually commensals. It goes around, not everybody. But it doesn't usually cause problems until it can get inside of a patient. And the problem with Pseudomonas is it's already antibiotic resistant.
Wow. So does that affect your lungs? Is it's safe to say because it's pseudo?
What was the other patients that worry about Pseudomonas infections?
Oh, AIDS patients maybe?
Not AIDS
There's
patients, they
other.
kind of worry about everything. But what is it? It could be Pseudomonas with AIDS, but there's another particular group that's not AIDS patients.
Older people?
No, it's
Geriatric?
older people.
Geriatric, I mean?
It's not a T cell response one. Cystic fibrosis. This is one of the main bacterial patients, bacterial infections in CF patients. And again, CF patients get a mucus buildup in their lungs, right? It gets nice and hospitable for Pseudomonas to get in there. Pseudomonas starts growing, and the problem is it's still already antibiotic resistant. So it's another double whammy for these guys. Now, what's Bordetella?
Whooping cough.
Pertussis, whooping cough. It is very virulent. It can cause disease in healthy individuals if they're exposed.
Even my baby over there, he got it when he was 10, and he had been vaccinated and everything.
And you got it?
Yeah.
Really?
Yeah.
But it's because I got vaccinated as a child. And
Yeah, but still, it should have.
But what do we say about vaccines, though? And that's the problem we talk about, that herd immunity.
You're not always getting the right, because it,
Well, that's the flu more so. With stuff like Bordetella and stuff, they're pretty narrowed it down. What do we? And this is the good time. We talk about anti-vaccine. Why should you be worried as a parent if this person doesn't want to vaccinate their child? If you vaccinated your child, why should anybody care? This is why people care. I'm just trying to point it out. I'm not stepping on anybody's views on anything. I already got. Hopefully we did that once semester, which I did not. But why do other parents worry about it? Why do they care?
Because of the different strains. I think that's what happened to my son, is that he was vaccinated properly, and I was always up to date, but he must have caught it from a different strain.
Not so much different strain necessarily. Is that what is the efficacy of vaccines? Are they all 100%? No, they range anywhere from 70 to 90%. That means if you have 100 kids in a school and they've all been vaccinated, 10 of them are not going to be protected. About 10 to 30 of them. So herd immunity, the idea behind that is you vaccinate as many as you can, the chances of anything getting in there are pretty slim. And that's why parents worry when other kids don't do it. And they've shown that. What have we had outbreaks of the last couple of years that we hadn't had in decades?
Well, you said the other day polio was coming back.
Polio, but it's not coming back. It's just stuck in a couple places. But what has come back in the U.S.? Measles and mumps. There have been outbreaks of measles and mumps. We have not had those in decades. So it shows that herd immunity thing works. Okay, what's Yersinia pestis?
The plague.
The plague, a very virulent disease. What's Francisella tularentia? Don't tell me rabbit fever. I know that's what it is. It is another incredibly virulent organism. It's not always lethal, but it can be. This is one of the ones they look at using as a bioweapon. It causes disease very easily in healthy individuals.
I thought you said that we couldn't get animal.
No, that's if it's a zoonotic disease, and that's a good point. Remember that, guys. If it's a zoonotic disease, that means it's an animal disease that can spread to humans, but usually humans are going to be dead and host for that. Because you can't pass it on to another person.
And that is not that? It's
No,
just called
no,
rabbit?
No. Rabbit fever. It's called tularentia. And it's not.
It was just called that.
Yeah.
Okay.
Well, because it's in rabbits a lot.
Oh.
And that's where they first sort of discovered it. And it's around here. My parents live in Albuquerque. They found 3 or 4 dead rabbits. And they take them to the health department. And they said the tularentia is there. That's one of the ones that they're looking at, some of the bioterrorists are looking at. So it's considered a potential bioterrorist weapon. All right, virulence factors and infectious agents. Extracellular enzymes are secreted by a pathogen, but these are not toxins, guys. They dissolve structural chemicals in the body. How do you keep cells attached to each other? You guys remember?
Bond?
Conjunction, you got gap junctions, that's communication. You've got tight junctions, you got desmosomes, and what do you have? The desmosomes have what, those calhedrons, you remember those? Kind of like little trailer hooks. They attach to each other. Some of these enzymes will dissolve those. They're not toxins, they're not killing any cells, they're dissolving structures. And they can help the pathogen maintain the infection, they can help it invade into deeper tissues, and avoid the body's defenses. Some of these other ones they secrete, actually we'll look at in a minute. So the bacteria will be in there, start secreting these enzymes, and it'll cause a clot to form around the bacteria. So what is that clot going to do to the bacteria?
Protect it.
Protect it and hide it from the immune system, right? So important to the virulence of the pathogen. Write this down, guys, and I want you to show this, because this is for all these different virulence factors. Mutant species that do not have some of these virulence factors become avirulent. Somehow we missed going over it, and I don't know how we missed it. But there's a strain that's an experiment that Griffith did, all right? It's called, so it looks at horizontal gene transfer. We were talking about being able to take DNA up. Did we go over that one in here? No, I know I missed it. So we talked about transformation, bacteria that can take DNA up from the environment, right? You all remember that? We didn't go over it very much. I need to go over it. So what Griffith did is he took a strain of strep pneumoniae that has a capsule. And the capsule is a virulence factor, right? That's the first one we've talked about. So we injected them. So here's some more mice we're going to take out, guys. Sorry about that. He injected some mice with the virulent strain of strep pneumoniae. What happened to mice? They died. Now there's an R strain. That's called the S strain. There's an R strain. The only difference is it does not make the capsule. Period. Everything else is exactly the same. It doesn't have the genes to make the capsule. You inject the mice. What happens? Nothing.
No, they stayed alive for a while.
They
and
stay
spread.
alive. Their immune system is able to go after the strep and kill it. So what they did is they took some of the S strain, the one that has the capsule, right, and killed it, and then injected that in the animal, and then injected the R strain in the animal. What happened to the animal?
He made antibodies against the S strain as well,
So you have the S strain, it's got the capsule, right,
but it's dead.
and you killed it, and you have the R strain, which is no capsule,
Living?
living. So this is heat killed. And this is live. You inject them both in the mouse, what happens to the mouse?
I think we already said no, so I don't know.
Still dies.
The mouse still died, they isolated the bacteria out from them, and all they had was the S strain.
And it was dead?
Yeah?
What?
So how did that happen? You did inject live S strain into, I'll put up the experiment for you
Okay.
guys, so you guys can see it better than I was drawing here. But the point is they injected heat-killed S strain and they injected live R strain, and when the mouse died and they recultured the bacteria out of it, it was all S strain. How is that possible? Hmm?
Oh, it's like you're saying the S was able to survive because it pulled the DNA from maybe the R?
They fought for the R, but there was still the S strain DNA floating around, right?
Right.
Because it killed it, you just heat killed it.
Right.
You still had all the parts, it was just dead. So it showed that through horizontal gene transformation, the R strain was able to take in the DNA from the S strain, and that S strain had the capsule, so now it converted the R strain to S strain.
Wow.
So that's, and they also done it in, have you done transformations in labs?
No.
No, it's kind of, you can also do it where they just take it, and we can do it in a test tube. Maybe we'll try and do some transformation at the end of the semester. We have a green fluorescent protein that when you hit it with a black light, it fluoresces green, and it's on a plasmid. We can try and transform it into these bacteria. So once it goes in, the bacteria should do what? They should fluoresce green when you hit them with the light. So we'll see if we can do that. They do it in a lot of labs. All right. But the point is, all that was missing from the R strain was what? I went a long way to get back to
The
here.
capsule.
Was the capsule, and so without the capsule, the bacteria is avirulent.
Wow.
All right, so here's what we talked about. Here's hydrogenase and collagenase. So I'm not going to have you memorize too much of these. But here's epithelial cells. Here's the bacterium. They release hydrogenase. It breaks apart those cell adhesion factors. Now you've got a what?
Opening.
Now you've got a gap between the two cells. Did it kill any of the epithelial cells? This in the
No.
No.
It just
Now
broke
it
the bond.
sneaks in here and releases collagenase, which allows it to get to the collagen and get inside of the
Cell.
tissue, the deeper bloodstream, the deeper tissue. But if this happens to be a blood vessel, what have you just done? You've gotten into the bloodstream, and salmonella can do this. Salmonella can get and become a systemic infection inside of you. Here you got another one here, it's got coagulase and kinases. The bacteria comes in, you release coagulase, it causes a clot to form around it. Now what? The bacteria can sneak around inside of you without being seen until it releases these kinases. Now it can escape and get back into the bloodstream. These are not toxins. Have they killed any cells?
No.
No. They've just allowed it to get in and sustain an infection. But if you got rid of hyaluronidase in this particular strain of bacteria, would it be able to invade the tissue? Yes or no? If I take the genes out to produce this enzyme, would that bacteria be able to invade anymore?
No.
No, it would be stuck in the GI tract.
Is that chemical used in our body to break other bonds or anything? Do we need that chemical?
We have enzymes that break down collagen.
But not that one.
Yeah, that's a bacterial one. I
Okay.
don't know the difference in all of them.
Oh, okay.
We do that one. All right. Toxins, on the other hand, are chemicals that harm or kill the tissues. Or they can trigger a host immune response that causes damage. We'll go back and talk about LPS again in a minute. Where do you find LPS? Lipopolysaccharide. Where in the gram negative?
Classroom.
Outer plasma membrane, good. All right. So, toxemia refers to, okay, I want to go over 3 different words. What does bacteremia mean? Okay, what does toxemia mean?
How much of the toxins are in the,
Just toxemia is just going to mean, emia at the end of things are going to mean it's present in the blood, right? So toxemia refers to the presence of a toxin in the bloodstream. So what is bacteremia?
how much bacteria in the blood?
Bacteria that's in the bloodstream, okay? And it's just going along for a ride. That's bad enough because it can go to other sites in the body and establish an infection. What is septicemia? It's not the same as being septic.
No, I was gonna ask.
Toxicity is what?
So which one? Septicemia? Septicemia is when the bacteria are actually growing, dividing, and metabolizing in the blood. So they can be secreting these enzymes and toxins and stuff like that. That's what leads to what's sepsis. And that's a huge problem you
Infection.
guys are going to come across when you guys get in hospital.
Infection in your blood.
Yeah, but what actually is sepsis itself, though? Somebody is septic. Is it the infection? It's usually caused by septicemia, but it can be caused by bacteremia too.
Oh,
What is it? Why is it so hard to treat? If it's just an infection, you should be able to pump them full of antibiotics, right?
that's
And
true.
they do. But does that work? Hmm. One of the leading causes of death in hospitals now is when people get septic. Sepsis is a massive immune response that's triggered by the septicemia or bacteremia. So it just goes, you have this massive super over immune response, which goes after it.
Oh, so it's like an allergic reaction kind of
No,
thing?
it's just not an allergic, it's not like massive, it's just, you've got bacteria in your bloodstream and colonizing organs and stuff. You just get this massive systemic immune response, and it starts going after the organs and everything else. It's an uncontrolled immune response. That's why it's so hard to stop.
I don't understand why that's bad though. Your body fighting it. Why would that
It
hurt
actually
you?
goes after you get multi-organ failure. It actually, it's such a huge uncontrolled response that actually starts attacking your own organs.
Oh,
So that's why it's so hard to get. They put you usually probably on steroids, I would guess. And, probably antibiotics to try and kill the bacteria. 2 types of toxins, 2 general classes, exotoxins and endotoxins. What is exo, what is endo?
inside. That's,
Exo, outside, endos, inside.
But
Does not mean they're gonna bring toxins inside of themselves, that would not be good. An exotoxin is actively secreted by the organism. So here you've got a bacteria and it's secreting an exotoxin, and this happens to be a cytotoxic one. What is it doing?
oh, destroying the cell.
It's killing the cells, right? Then, and this one, the only endotoxin we're gonna talk about is LPS. So dead gram-negative bacteria release endotoxin, the lipid A from LPS, which induces its effects such as fever, inflammation, diarrhea, shock, systemic blood coagulation. But here you got a bacteria, it's been phagocytosed by a macrophage. Macrophage chews it up, exocytoses out what's left of it, but what comes out? Lipid A. Lipid A gets to the blood vessel and cause all these
Oh,
problems. Or you just have a bacteria that dies, as soon as it dies, it starts releasing lipid A. So
oh,
that's how all gram-negative bacteria are potentially what?
fatal.
Not fatal, but pathogenic. All right, so let's look over this. Let's go pretty good on time here. Exotoxins, mainly gram positive, gram negative. Who's left? I mean, you got some other ones out there, that's kind of weird. Right? Metabolic products secreted from a living cell, these are gonna be proteins or small peptides. Toxicity is what? What is the most potent toxin known to man? In terms of how toxic it is, how much it takes to cause death.
That's what I was
Radiation?
thinking.
Anybody know?
Are we thinking chemical? Like, what are we talking here?
So, well, anything, I want you guys to tell me, this is my natural, it can't be bad for you. Right? Botulinum toxin is the most toxic,
Oh, wow.
most potent toxin known to man, and it's secreted by Clostridium botulinum, also known as what? In the
Botox?
cosmetic industry.
Botox?
Botox. So I'm gonna inject the most potent toxin known to man into my face. Well, we thought about that a little bit strange, don't you think?
So why don't they get affected when they get it? I've always wondered that.
It's because they use such small doses of it and everything. The first guy who invented Botox, they had one of those big Erlenmeyer flasks, they were swishing it around, it was full of Botox. There's probably nothing that thing would kill the whole planet, would be my guess. All right, high toxicity, they're not heat stable because why?
They have to live at our body temperature.
No, this is just the toxin itself.
Oh,
Why aren't they heat stable? Above 60 degrees centigrade, which is also what they pasteurize.
because it breaks down in protein like this.
They're protein. If they're protein, they're going to be what? Denatured by heat.
Yeah.
So that's a good thing. You can cook most of your food and get rid of these things. Unless you go to Chipotle, in which case they might. Effect on the host is variable, depends on the source. Cytotoxic, neurotoxic, enterotoxic is your gut. Nephrotoxic would be what? Kidneys. Hepatotoxic would be what?
Liver.
Liver. Fever producing, no. These are toxins that kill cells. Antigenicity, which means, what is antigenicity? It's almost the same as immunogenicity.
That you make the antigen?
Nope.
Ah,
It says strong stimulates anti-toxin antibody production. If you are very antigenic or high antigenicity or high immunogenicity, you stimulate the immune system. Since you can do that, you can make what? Toxoid formation for immunization. If it's got high antigenicity, you should be able to make a what for it? This
A
is not the
vaccine?
bacteria, this is the toxin itself. What can you make if it's highly antigenic? Or highly immunogenic is probably a better word. You can make vaccines, you can make antitoxins. They have antitoxins for botulism, for tetanus, for snake bites. Okay? Representative diseases are going to be botulism, I just talked about, tetanus, gas gangrene, which is pharyngitis, diphtheria, cholera, plague. You better hurry up and get that one, otherwise you're a goner. Staphylococcal food poisoning. Now let's look at endotoxins. Endotoxins only come from where?
gram-negative.
Gram-negative, and they're where? Portion of the outer cell membrane you were talking about, in particular the lipid A part of LPS. Low, so toxicity is low, but if you get a whole bunch of it,
Fatal.
it could be fatal. So if you had a whole ton of gram-negative bacteria inside of you that all died at the same time, what would happen? That could be bad, because you can release all this lipid A. And this actually happens, it happens with some parasites too, right? If your dog has heartworm, what's the treatment for heartworm?
Oh.
Kind of going off the other tangent here, but it has to do with the immune system, and this is what I'm talking about. What is the treatment for it? Anybody know? Do they want to kill all the worms right at once?
No,
Why not?
you need to release all that.
You release all that dead worm from the inside, and you have this huge immune response, and you're basically anaphylactic shock.
Oh.
Same thing can happen in this case. It's
So-
not a protein, it's a lipid. A lipid's very delabile. Can you destroy lipids by heating them up? Not really. So look at it here. Where is it?
Stable.
stable for up to 1 hour at an autoclave
Wow.
temperature at 121 degrees centigrade. So that's the problem, you can't get rid of them. Fever, lethargy, malaise, shock, blood coagulation, and a couple others can cause systemic shock. Is it fever producing? Yes, why?
Your body's immune response.
Because it's an immune response, that's what it does, it triggers an immune response that goes at you. It's not directly toxic, it's indirectly toxic. It's a lipid, lipids make really poor antigens, so you can't make a vaccine against it, okay? And you have typhoid fever, tularemia, toxic shock, urinary tract infection, meningococcal meningitis, who knows, anything that's gram negative that could grow inside you potentially could be what? Pathogenic, right? All right, let's get through these, I'll let you guys go today. What are antiphagocytic factors? What's phagocytosis?
To eat it.
The inability to eat.
Yeah, it's got a phagocytosis to eat, so your macrophages, your neutrophils, your dendritic cells, they all go crawling around your body looking for bacteria, and when they find them, what do?
Eat it.
They grab them, eat them, pull them in, and break them down. So factors that prevent phagocytosis. We already know about the main one, what is it? The capsule, okay? Capsule has 2 jobs. What are the 2 jobs of the capsule?
Protection and
Protection, so composed of chemicals that are not recognized as foreign and slippery and difficult for phagocytosis to engulf, for phagocytes to engulf. 2 things it does. A, it's stealth, it's camouflage. It helps hide from the immune system. Got a bunch of glycoprotein stuff on there, the immune system kind of looks at it and goes, I don't know what this is, I'm not worried about it. If it does detect it, it's like trying to grab onto what?
like Jello, like
It's a really slippery thing. They used to do, back when my parents used to grow up, they used to get a 4th of July and get a watermelon, then grease the watermelon up, throw it in the pool, and all the kids would jump in and try and get the watermelon out of the pool. Are you ever gonna be able to do that? No, so that's kind of like what's going on here, right? Guess they don't do that much anymore. Kids did it all the time. I guess no video games back in the day, huh?
yeah.
Antiphagocytic chemicals, okay? This is going to prevent fusion of the lysosome and the phagocyte vesicle. Leukocytes directly destroy phagocytic white blood cells. 2 different things they can do here. Let's go after this. Once something is phagocytosed, what happens? It gets drawn inside.
Mm-hmm.
Now what's going to happen? But remember from shipping over this in 1306 a little bit.
Breaks it down and then spits it out.
So it's got brought inside your phagosome, usable bacteria. Okay. What's going to happen next? The nucleus in there.
Breaks it up
How does it break it up?
with those little.
A lysosome is going to fuse with it. A lysosome is going to fuse, and you're going to get called a phagolysosome, and it's going to dump all of its all its digestive enzymes in there, and ideally it's going to do what?
Disintegrate it.
Disintegrate that out. If it happens to be gram negative, it might put a little LPS out here, which would do that. If it's gram positive, you don't worry about that, but what they're talking about here, Neisseria gonorrhea can do this, I think meningitis can do this, Listeria can do it.
Uh-oh.
Tuberculosis.
Tuberculosis?
Yeah.
It can do, yeah, it gets the bacteria. Does it prevent lysosome fusion?
Yeah, it forms like granulomas or something like that. It's
Yeah,
like-
does the tuberculose?
Yeah.
So, yeah, the thing is, I guess it does prevent lysosome fusion. So several of these organisms, once they get inside, if they can prevent the lysosome from fusing with the phagosome, I know that's a lot of somes going on here, what's gonna happen? What happens?
Oh, can they take over the cell?
Do they get killed?
No.
No. Not only do they get killed, they can start to multiply in there. And not only can they multiply, they're also hiding from what?
Your body, your
The
immune
rest of the immune
system.
system. They're actually in the cell the immune system is supposed to kill it. So
Wow.
they can do that. So that's what they're talking about there.
What did you say can do that?
TB can do it. I know some of the Neisseria can do it. Listeria, I don't think actually gets in the phagosome. I don't think. I need to look. But we'll go through each one of those later
That's
on.
cool.
We'll talk about it. And then leukocytes, which is what? They're killing the white blood cells that are designed to go after them. Very good. Yeah, that's not gonna work. All right, okay, we'll go ahead and let you guys go now. All right, so no lab today, right?
Right.
You have an online lab. I'm going to see you guys do it. It's a pretty interesting lab, I think. It'll take you guys through PCR.
Can you possibly post the link of the video so we can finish watching it?
Which one?
The video we watched in lab last time?
The mosquito?
Yeah.